Okay. I'm only waiting for Professor McCaffrey. I hope he pushes the right button. He says, got it. Yes, thank you very much. Okay. Ladies and gentlemen, good morning. Good morning, Miss Siyu Wu. Welcome. Hereby, I open this academic ceremony in which Siyu Wu will defend her academic thesis entitled Modulation of Myelin Phagotosis by means of anti-inflammatory treatment as a therapy of spinal cord injury. May I invite you to present a summary of your thesis in the next 50 minutes and couple to this the conclusions of your thesis, and I would like to give you the word. Ms. Wu.
Thank you. Thank you. So, dear pro-reactor, dear members of Corona, dear families, friends, and audience, thank you for attending my oral defense. It's my honor to present my thesis here. My topic is modulation of myelin phagotosis by means of anti-inflammatory treatment as a therapy of spinal cord injury. So, spinal cord injury is a completely or partially damage to the spinal cord. It causes the loss of sensory functions and of voluntary control below the injury site. Worldwide, the incidence of SCI is 113 per million per year. Over twin cells, SCI cases are high energy traumas, which are caused by force, traffic sports, violence, or force. The rest are disease-related, called non-traumatic injury, such as spinal stenosis, infection, and compression by tumors. According to the time course and mechanisms, spinal cord injury distinguishes between primary and secondary degeneration. First phase includes blood spinal barriers disruption, nerve anchors, and myelin breaking down. Secondary injury happens in later days to months. It's a series of immunoresponse. In this phase, myelin debris accumulate in the lesion site and exacerbates the inflammation. That consequence an obstacle for anxious regeneration. Inflammation causes the most devastating effects. There are two arms in my thesis. First, remove myelin debris to reduce inflammation. Second, use anti-inflammation methods to improve SCI recovery. To accomplish our goals, we provide three strategies. First, we use the chemical compounds to enhance myelin clearance. Retinoic acid is a ligand for RAR receptor, which is involved with the remyelination and also constitute an anti-inflammatory feedback in nervous systems. We also choose the bile acid as the therapy for spinal cord injury. Bile acids are related to the lipid metabolism and also have anti-inflammatory effects in the CNS. Secondary is the cellular therapy. A promising approach for spinal cord injury is stem cell implantation. Bone marrow-derived stem cells prepared from fresh human without extension in vitro was used in our research. That cells provided by Neuroplast company. The third therapy is the combinatory therapy, which is used the bile acids and the stem cells at the same time. For the outcomes, part 1 in my thesis, we wanted to know how retinoic acid affects myelin clearance. To quantify the myelin phagotosis in culture, we use a lipophilic tracer called DIO to label myelin, which could be observed and quantified by cytometry and microscopy. an antibody specifically against the myelin basic protein to confer this uptake. pH-sensitive dye called pH rhodo was also used to label myelin and show a dynamic change about the myelin uptake process, like the video shows there. That also could be quantified by a microplate reader simply. From the figures, retinoic acid enhances myelin phagotosis by macrophages from 29% to the 37%. It was enhanced. So what's the mechanism involved in this study? We measure several myelination-related receptors in macrophages. The cell such as the CR3, CD36, TREM2, transglutaminase 2. Our S2, which is the formation of the phagototic cup, and also a target gene of RAR receptor. That's the results. The expression of transglutaminase 2's mRNA and protein also significantly overexpressed after the retinoic acid treated. When we use transglutaminase 2's inhibitor to abolish this increase, we found that the phagotosis level goes down, like shows there. We also interested in the interactions between myelin phagotosis and the inflammatory phenotypes of the macrophages. Retinoic acid significantly reduces the expression of the pro-inflammatory factors like INOS and IL-1 beta's expression. That's part 2. In spinal cord injury models, the lesion site is under inflammatory stress. We wanted to know if the compounds could also help improve myelin phagotosis under this inflammatory stress. Myelin labeled with pH rhodo, phagotosis level was reflected by the fluorescence index. We found the myelin phagotosis decrease when the cells exposed to the LPS or infrared gamma. When we inhibit the inflammation by NF-kappa B pathways, use the inhibitor called BAY1142, the decrease was disappear. We test 3 kinds of bile acid affection in the inflammatory conditions of the myelin phagotosis process. Only TLCA rescue the myelin phagotosis under inflammatory stress. To explain this affection, we do several tests as the part 1. We found a key point called GAS6, means growth arrest specific 6, which show to interact with myelin phosphorylated series and the macrophage's surface receptors. NF-kappa B pathways active the inhibit GAS6 expression and secretion. TLCA rescue the GAS6 expression. Studies in part 3 are about the therapy for spinal cord injury animals. In this part, we implant the bone marrow stem cells into the ras cisterna magna, like shows in the figure. So BBB is a standard assay to evaluate spinal cord injury animals' motor function. Besides the SCI negative treated group, we also cite a misoprostol inject group as a control group, which is always for the clinical treatment. We found a significant motor functions recovery improve in BMSC cells group compared with the mesopenicillin treated group in first 5 weeks. When we conduct further analysis, we observed that BMSC treated animals' microglia activities in the lesion center was reduced. The histological results also showed less axonal degeneration after BMSC is treated. In part 4, we investigated the combinatory therapies. Tuscas was one kind of the bile acid in the in vitro experiment. Tuscas was injected to the animals after spinal cord injury. We found that animals have a better recovery of motor functions in the first 4 days with Tusca treated than the control group, which I didn't show there. But another interesting results is about the autonomic functions called the euro retentions. Animals lost detractor muscles control after spinal cord injury that causes euro retention. From the figures, Tuscas treated animals getting neural functions recovery earlier than the control group. It started at the day 2. So, but Tuscas communicated with the bone marrow stem cells didn't get an additional benefit compared with only Tuscas alone treated. The inflammatory cytokines expressed was also reduced by the Tuscas treated group, and we also found the macrophages activities was reduced after treated with Tuscas. So for conclusion, we got three statements. So first is anti-inflammatory treatments, retinoic acid and taurololic acid enhance myelin phagotosis. Second, bone marrow derived stem cells has the neuroprotective properties after spinal cord injury and reduce the axonal degeneration and inflammation. Third one, the combinatorial therapy doesn't provide an additional therapeutic benefits for the motor functions recovery after spinal cord injury. Thank you for your attention. I give the word back to the pro-reactor. Thank you.
Thank you very much, Ms. Wu. Maybe you can take down the screen. Yes, and okay. We have full screen. Thank you very much, and for your clear presentation, very nicely illustrated, very clearly presented. But we're now going to start your opposition. And the opposition will be opened, not by the chairman, but a member of the assessment committee, Professor Prekarts, who is also the secretary of our meeting today. And I would like gladly to give Professor Prekarts the opportunity to start with you a first discussion. Professor Prekarts.
Thank you, Mr. Pro-Rector. Dear candidate, first I would like to congratulate you with this very nice thesis. I also would like to congratulate your supervisors, who I know well and who are experts in this field. I really enjoyed reading your thesis and learned a lot. The multidisciplinary experiments must have been a tremendous lot of work, I think, and especially chapter 4 and 5. That's a lot of work. I also appreciated the valorization component of the work, as the bone marrow derived stem cells preparation you tested in chapter 4 and 5 are intellectual property of the company Neuroplast. So your thesis, your work is a good example of a fruitful collaboration between academia and biotech. So all in all, many compliments. Nevertheless, I do have some questions which I would like to discuss with you. So in particular related to chapter 5. Interestingly, this chapter was greatly introduced in the last sentence of chapter 4. So I'm going to read that sentence. The intrathecal transplantation of human bone marrow-derived cells prepared via negative selection and without cultivation will contribute to a combinational therapy of spinal cord injury. So that's it's an announcement. So I was looking forward to chapter 5. However, the results of chapter 5 do not suggest this, or at least indicate that a combination of bone marrow-derived stem cells with the bile acid TUDCA, do I say it right? TUDCA, okay.
Yeah, TUDCA.
It's not, or TUDCA is not that successful. So first, an easy question to kick off. Also for myself to get a better picture, what is TUDCA? I did some Googling and I also checked the company where you bought it. And actually it is described as, I will read it, a detergent useful for the solubilization of lipids and membrane-bound proteins. So I thought, is this useful? Can you help me a little bit why you chose this bile acid?
Okay, thank you for your kind words and thank you for your question. So what's the TUSCAS? TUSCAS, the full name of TUSCAS is Turorous or the deoxynucleic acid. Is kind of bile acids is compounded by the humans. Is not a natural bile acid. So I choose 3 kind of bile acid, or at least in the part of in the chapter 3, at least 3 in the in vitro experiment. They're called TLCA has a better results than the other, but for the in vivo experiment, I choose the TUDCA or the treatment for the in vivo experiment. The reason is because the TLCA has some risks, to cause the liver disease in animals. So we use a lower affinities bile acid to as a treatment for the spinal cord injury. The bile acid is synthesis from the livers, so that will have an anti-inflammatory effect for anti-inflammatory effect in the livers and also in the nervous systems. There have some reports for this. TUSCAS have the anti-inflammatory effects in nervous systems.
Okay, yeah, it's clear to me that you did your homework. Yeah, it's actually coming more from the traditional Asian medicine, I think. Yeah? I saw this from the liver of bears, and I've always a bit of a plant extract feeling. You know, it can do a lot of things, and I also checked that it's anti-apoptotic, anti-inflammation, good for lipid metabolism. Actually, I also saw that you can buy it at Amazon as a supplement, and it's good for your liver, as they say. It's amazing. It seems to be very promising. Good. I have other questions related to chapter 5 mainly. So when I saw figure 3 in that chapter at page 167 of the printed this version, so page 167, figure 3, I realized I had seen that figure before. And if you go back 2 pages, you see it again. So this is, I had seen it actually in figure 2 at page 165. So I'm afraid you made a little copy-paste error, because figure 2 should partly deal with bladder function and not locomotor function. But I'm happy that you showed in the presentation those figures of bladder function. So thank you for that. Anyway, in figure 3a, you made the point that T600, that's 2 times 300 milligram per kilogram of TUDCA, might have some beneficial effects on motor function. I agree. Then I go to figure 3b. I see the figure, I actually think there is not much. And then you make the statement in the text, combinational therapy with TUDCA and bone marrow-derived stem cells achieved higher BBB scores between 1 and 6 weeks, while these differences did not reach significance. Can you please explain this strange sentence to me? Because this is really, there's a difference, but it's not significance. What is this? Can you, what is this? So it's a, if you go, I will help you. So you have figure 3, and if you go to the left on page 66, so that's the first, and the bottom, you see in the middle somewhere at the end of that paragraph, combinational therapy with TUDCA and bone marrow stem cells achieve higher BBB scores, while these differences did not reach significance. So I'm a bit lost. Is it different or is it not different?
It's not a difference in the later weeks, but in the first 4 days, they have a significant difference between the TUDCA treatment and the control group. But for the combination of the bone marrow stem cells, I don't have a significant difference between these two treatments.
Okay. Yeah, I was a bit lost because in the abstract you don't sell it as an effect, but here you try to, at least to me, try to say, yeah, it's not statistically significant, but there is a difference. In my view, something is not different if there's no statistical evidence for that. Anyway, I have an add-on question to figure 3A. We go back to the top panel. There, actually, I missed the combination group. So, which is shown in figure B with the nice blue squares. What didn't you test in the first week this group as well on BBB score? Why is it not in the figure? That's what I mean.
Sorry, I didn't hear you clearly. So why I didn't test this in first weeks?
Yes, the combination group.
The combination groups. Yes, I do test the BBB scores in the first weeks, but it's not shown there because that's not that experiment that surgery not start at the same times. I mean, first I only do the surgery, do the SCI experiment with the TUDCA treated. That's at the first experiment, first step of the statement. So we want to find the TUDCAs that have any effect, have any benefits, compare with the control group. So after we got positive results, the TUDCAs have a little improve of the recovery compared with the control group. So we do the next step is use the TUDCAs and stem cells or the combination therapies. It's not at the same time. So sometimes it's because of the ability of the animals, it's a little hard to compare or put the lines there.
Okay, so so these experiments were not done at the same time?
No.
These experimental groups were not tested all at the same time. That's what I mean.
Yeah, not at always at the same times.
Always at the same time.
No, not always.
Okay, then I understand. Okay, then I understand. But still, I think you could have tested the combination therapy, not only. Okay, indeed. Now I see in B it's there actually, but it should have been also depicted in A. I think this combination group to give more clarity. Anyway, I have some more mechanistic questions to follow on because I was also probably like you and the company too a bit disappointed because there's a lack of this additive effect between these two therapies. So what could be the explanation for that? I gave it some thoughts. I read your discussion in chapter 5 with great interest, but I missed some possibilities, I think. So in chapter 3, you showed that TUDCA needs LPS, so an inflammation more or less, to phagocytose myelin, otherwise it doesn't phagocytose myelin. And what do you think about the explanation that bone marrow-derived stem cells dampen inflammation just enough so TUDCA does not work anymore that optimally? Because this bile acid needs inflammation actually to work properly to phagocytose myelin, if you think that's the major readout.
Okay, thank you. First in the part 3, when I only treated the spinal cord injury models with the bone marrow stem cells, actually we only have a tiny difference between the bone marrow stem cells treated group we compare with the control group. For the motor functions, it's not a big improve, but when we analysis the tissues or with the mRNA levels in the protein levels, we found that the bone marrow stem cells do have could reduce the inflammatory response. But that happens for 5 weeks, I think, is lasting for long terms. The tooth cast is treated only in the acute phase after spinal cord injury models. It's only got at most 5 shots per day, 1 shot per day for 5 days. So maybe the tooth cast only have the anti-effect or have the benefits in the first 4 to 5 days in the acute phase.
Thank you. It's a good explanation, actually, which you give here. Thank you for that. I have another possibility. This is my last question, also related to this. So a second mechanism that crossed my mind has to do with microglia. So based on the literature, you stated in chapter 3, I think, that inflammation or LPS stimulation raises the proportion of microglia that phagocytose, so it activates them. This appears actually contradictory to macrophages, of which you show in chapter 3 that LPS stimulation or inflammation reduces their phagocytosis capacity for myelin in this case. Could it not be the case that the effects of microglia and macrophages cancel each other out? So in the end, this explains a lack of efficacy of what you see in your study. What do you think about that?
Okay, so actually, when I started the experiment in vitro for the myelin phagotosis, I tried both of these cells, the macrophages and the microglia. Yeah, we do have some experiment to show the microglia also have enhanced the phagotosis of the latent bees, latex bees, but it's not for the myelin debris. So that's the reason why we only in the later experiment we only have the macrophages or the main force. We only detect the macrophages phagotosis of myelins. But it's a little that's for the in vitro experiment. For the spinal cord injury models for the animals is a conflict. Macrophages and microglia are the conceptors help each other to do this phagotosis activities.
I'm satisfied with your answer. You don't think that's an explanation, and that it could be actually followed up a bit more in vitro with experiments. I thank you very much for your answer, and I give the word back to the prorector. Thank you.
Thank you very much, Professor Prikkaerts. And the opposition will be immediately followed by Professor McCaffrey. He was a member of your assessment committee, and he is joining us from Aberdeen. And I would like to thank him that he has the time to join us today. Professor McCaffrey, may I give you the word for your opposition?
Well, thank you very much for that, Prorector. And I'd also like to join Professor Prikkaerts in his comments regarding your thesis. I thought it was excellent and a tremendous amount of work done. And I really enjoyed the combination of both molecular work as well as the physiology and behavioral studies on the rats. And I really do, although I mainly do the molecular work myself, I understand completely the amount of work involved with the rats. So I'd like to start off my questions with a general one about your understanding of secondary degeneration. What I'd like to actually get is your views on why there is such secondary degeneration. In other words, why would it be an evolutionary advantage, or it seems like a disadvantage, to have a system that actually results in more damage to the spinal cord rather than try and repair the spinal cord and get it to function again. What are your thoughts on that?
Okay, thank you for your questions. So just to make sure I got your questions correctly, your general question is why I think the anti-inflammatory response is important than the repair of the spinal cord injury in the Lyran center or not?
Yes. How did it evolve that you have a system that actually causes more damage rather than tries to repair the spinal cord?
Okay. So in my studies, I more focus on the myelin degeneration, and that's also the same conditions or the other cellular debris. After the first injury, is chemical damage to the tissue, to the spinal cord, that cause the degeneration or the breaking down of the entrance, and also for the myelin is surrounded part of the anxious, and also called the cell breaking down, that will release a lot of cell members and the containers of the cells that will stimulate the inflammatory response, which will accumulate many macrophages and microglia in the Lyran site. If we don't control that activities, they will have too much macrophages or microglia in this Lyran center and do a lot of uptakes that will cause the damage much larger in the Lyran site. They will get a hole in the spinal cord. So that's the reason why we want to do the cleanings. For the repair, also after the primary injury, the secondary injury also include the spinal scar and the spinal scars systems that will be an obstacle for the repair of the neural cells in the Lyran site. Yeah, I think that's what's in my mind.
So what, as I say, you would have thought evolution would have led mammals to have a system that could repair, but that's not the case. More damage seems to be occurring. So I suppose the answer to my question is that at this point it isn't understood, and although I think that is important to understand, because what you're trying to do is trying to either reverse some of those systems or encourage the systems that are trying to repair. My second question is regarding, you state a number of times in your thesis how the bile acids that you're employing have been used in traditional Chinese medicine. And I'm very interested in your view of Chinese medicine. Do you think there's a lot of promise there in the sorts of compounds that have been used in Chinese medicine, that there's a lot of promise, or do you think some of these compounds are too complex to be used? So let me know some of your thoughts on that.
Okay, thank you. So for the Chinese traditional medicines, so for in my opinion, I trust for the Chinese traditional medicines, but it's very completely compounds of this. For the actually in the Chinese traditional medicines, when they use the bile acid, they use the natural product from the animals. They use this for the treatment of the disease. But in our research field, when we do the research, we will analyze the chemical compounds of the bile acid. So we just catch up the most important efficiency part of the medicines to do the research. So that's the important thing. We must know the clearance, know the compounds of the chemical compounds, know the chemical structures of the compounds. I think that is important. If we could do this always, all the time, I think the traditional, the Chinese traditional medicine will have a promising future in the science field.
Right, thank you. That's a good answer. So I'd like to ask some more perhaps technical questions. In chapter 2, you had a very interesting series of studies, and you used a purified rat myelin, and that was purified from normal rats.
Yes.
And I wonder what differences you think you might have if you use myelin that had been prepared from the injured spinal cord.
Yes, thank you for your question. It's really an interesting question. So, actually, I didn't consider these things before. Yes, I think the myelin is not a purified protein or lipid things. It's a makeup of protein and fatty substance. It's already a complete peptide. It's not specifically protein or lipid, these things. So I think it's not a very big difference between the normal rats and the spinal cord injury rats. I think it's the same thing, or it's almost the same thing.
Yeah, it's a tough question. I think that's a quite reasonable answer. I would like to see if your answer was correct or not, and actually see the results of the experiment. I could imagine a number of reasons. I'm sure you could yourself as well, why there could be some differences for various reasons, that the myelin might be modified, or there may be, you never really completely purify these compounds, and so there may be other compounds that carry over. But anyway, I liked your answer. My next question is because you may know or not, I do retinoid research, and so that's my particular interest. And so I'm going to ask you a question along those lines. So you're using the ligand bexarotene as the RXR agonist, and a lot of your thoughts in your thesis were along the lines that this ligand would activate RXR and heterodimerize with RAR, so activate the retinoid system. Are there any reasons why there may be other effects of an RXR ligand?
Okay. Thank you. So I choose all trans retinoic acid, or the agonist for RAR receptor, or I know, or I searched from the other reports. There have another retinoic acid is called niacin retinoic acid. It's agonist for both RXR and RAR receptor. So we choose all trans retinoic acid that is activator only for RAR receptor. So we more focus on the RAR receptor instead of RXR.
Yes, no, I understand that. Let me ask a bit differently. What do you understand about permissive versus non-permissive actions of RXR? And I know actually I've discussed this with York in the past. And this isn't always so clear, but what do you understand about this concept of being permissive versus non-permissive in terms of an action of RXR?
RXR.
Yeah, so in other words, in some cases, RXR heterodimerizes with another nuclear partner, for instance, RAR, but
FXR.
from what dogma says, a activator of RXR won't affect the system. But some other partners of RXR, like PPAR, for instance, and I think FXR as well. Yes, you go on, yes.
Yes, actually in my seasons in the experiments, I also test some of the other partner of RXR, like PPAR alpha, FXR, these receptors. I do have some agonist for these receptors, and also I wanted to measure the phagotosis levels. Goes up or goes down. Unfortunately, I didn't got a positive results with the other agonists, with other ligands. Retinoic acid, ultras retinoic acid is the only one we found that they have that increase effect.
Right. Okay. Yeah. That's lovely. Thank you very much for your answers to my questions, and I'll leave it there for now and hand you back to the prorector.
Thank you very much, Professor McCaffrey. And the opposition will now be followed by Professor Zimmermann. Professor Zimmermann, may I give you the word?
Yes, thank you, Mr. Prorector. Dear candidate, also I want to congratulate you with your nice thesis and this combination of work, of in vitro work and animal work from different aspects, with, I think, a direct clinical implication possible. And also your work is clearly a collaboration between different institutions, and you worked in different places as well, which is very nice to mention as well. And also, of course, congratulations to your supervising team. But as you know, we are here to ask questions, as the others have done. So I'll go to this clinical application first. What is needed to bring, let's say, the three treatments you studied, the retinoic acid, the bile acids, and the stem cells, to clinical trials and clinical application? The easiest to answer probably is the stem cells, because that's already in phase 1 trials. But let's start with the retinoic acid. At the end of the chapter, that's chapter 2, you say that clinical investigations of retinoic acid are needed. But are we ready for that? Because what you showed was mainly in vitro studies. It's not even animal studies in this chapter 2. So are we ready to go to clinical trials for retinoic acid? And if not, what is needed? If yes, why?
Thank you for your questions. So for the retinoic acid, actually they already have some more preclinical trials for the spinal cord injury. They have some more reports in 2016. I think it's Thomas from London. He published a report about the retinoic acid for clinical spinal cord injuries treatment, or the therapy, or the treatment. So in that papers, he demonstrates that the retinoic acid have some benefits for the nervous growth and nervous growth in the CNS and also in the PNS. They already have some experiment about these things. So Yeah, it's also because of my PhD seasons is limited, so it's not too many times for me to test the retinoic acid.
No, that's fine. You have done good work. It's not a critique.
They already have some positive effect.
Yeah. So let's go to the bile acids. At the end of the discussion, you write, in order to provide a regenerative therapy to patients suffering from spinal cord injury, it will be crucial to disentangle the differential effects of endogenous and synthetic bile acids on intracellular pathways. And also your results are not completely promising, also with the different bile acids. So I think bile acids are not ready for clinical use, is it? But there are some animal studies, of course, there. But so what would be needed?
Okay, thank you for the bile acid. Or the I know the bile acid. No, especially to the TUSCA, have 5 reports for the spinal cord injuries recovery in the SCI animal models. They have some more positive. They have a more positive than us, the better recovery of the animal models. The TUSCAs, the TUSCAs in animal models maybe have a promising benefits, but it's not enough for the clinical trial, because there's only several preclinical trials. Mostly they said the TUSCAs is have the benefits, but actually in our studies, the TUSCAs only have the positive effect in the early phase, in the first 4 days, not last for long terms. So I don't know, maybe it's not ready for the clinical trials.
But of course, the advantage is that it doesn't have much side effects, although you did mention that this TLCA, this other one, did also give some lot of problems. But let's go to the stem cells, because that's, of course, the most promising, let's say, and also there are some phase 1 trials and so going on. But you mentioned somewhere, let me first ask that so there is a lot of small animal studies, but not so much large animal studies on this spinal cord injury. And you also say somewhere, I don't remember where exactly, but somewhere in the discussion, that it's also not needed to go to large animal studies first, because you use probably own cells and so. Is that so? Because I would think to go for clinical trials, I would first go from small animals to large animal studies to prove the concept, and then go to clinical trials.
Yes, thank you. So the mostly used animal models, the model animals is the mouse, but for the spinal cord injury, sometimes is always be the rat, because it's a larger bodies. It's easy for the surgery to for the researchers to do the surgery in the spinal cord. But even we switch this research to the big animals like the monkey or the other human being-like animals, the final the final goals is we wanted to use the stem cells in the human beings. So I think if they have enough, they have enough positive results, so maybe we could try to switch this trial on the human beings, because that stem cells is from the human beings, it's not from the model animals.
Yeah. You also mentioned that it's quite easy probably to use the own cells of the own patients, like hematopoietic or bone marrow stem cells, I should say, from the own patients, and not necessarily from a donor. But of course, in this company now, they use like a donor, and you use it in the animals. So probably you could do it from the own patient, but then it's not like if you use it from the own patient, you always have to go through the processing, and it might be maybe not as safe. And I don't know if there would be any rejection anyway from these mesenchymal stem cells, even from a donor. So why would you, in principle, it would be safer to use their own cells, but do you think it's necessary to use own cells, or you could also use from a donor if you purify them enough?
Yeah, yes. I think it's better the stem cells from the own patients. It's better, as we know, it's better have some advantages without any immuno reflect like these
I
things.
realize that, but it's also more difficult if you want to do this all over the world. Then everybody has to start isolating these cells according to this procedure of the current company. While if you use it as a commercial product from donors, it's much easier.
Ms. Wu,
Yeah.
could you please give a short answer, because we have to proceed.
Oh, okay, thank you. A short answer. Yeah, but that's our purpose. We more focus on the long term, the chronic stage recovery. So so we have times to prepare the stem cells from the patients.
That's true. That's true.
Yeah.
Thank you very much. Thank you for your answers. I'm very happy, and I give the word back to the professor.
Thank you very much, Professor Zimmerman. And I would like to continue swiftly with Dr. Panis. She's affiliated with the Department of Pediatrics at the University Medical Center, and I would like to give her the word. You have to unmute, Dr. Panis.
Thank you, Mr. Prorector. Dear Siyu and supervisors, congratulations with this work, and thank you for performing this incredible work and study and making treatment for these patients possible in the future. I would like to ask you about methylprednisolone. As a clinician, I follow protocols, and in case of a patient with spinal cord injury, I do not treat him with methylprednisolone because evidence is lacking for better outcome. I would like to ask you, why did you compare your treatments with methylprednisolone?
Okay, thank you for your questions. So my answer is the reason for us to cite methylprednisolone group as a control group is because the methylprednisolone is the licensed clinical treatment. Yes, I think that's the only licensed drugs for spinal cord injuries. So I think the methylprednisolone is started at research in 1980s. That's the first use of the methylprednisolone, and later, maybe in 10 years or 15 years later, the methylprednisolone get the license for the NACSS. They got the promise to use in the clinical treatment. But later in the recent reports, we found the methylprednisolone have some more side effects, not benefit for spinal cord injury. So we, for the efficiency, they didn't suggest to use this, but it's still the clinical treatment. So we said this control group to have some evidence to this.
Okay, thank you for your answer. Then I would like to go on to your neurocultures. I was wondering, did you culture them yourself? And if not, what were the problems or the things you have to solve using them?
Sorry, I didn't got your questions.
Your cultures, your myelin cultures, did you culture them yourself?
For the myelins?
Yes.
Yes. Yes, I insulated the myelins myself from the right brains. I do this myself.
Yeah. You cultured your cells yourself.
Yes.
Okay. Okay,
yes.
That's fine. Then about proposition number 6 it is. Do you have them with you? Maybe you can.
Yeah.
Maybe you can read out that.
Yeah, number 6. The pH-sensitive pH rhododyes real-time that called label lipid debris gives a more efficiency and dynamic assessment of Phagotosis process. Is this one, the number 6?
No, about the pH rhododyes assay.
Yeah.
Do you know which proposition I mean?
Sure.
It's I will read out. The pH rhododyes assay of myelin phagocytosis give a more efficient and dynamic assessment of anesthesia. I have a question about that.
Okay.
Yeah. As a pediatrician, as a metabolic pediatrician, I'm interested in lysosomes and the activation of phagocytosis. But phagocytosis is an energy-demanding process, and because the lysosomes they only work in an acidic environment. Hydrolysis breakdown myelin will happen in acidic environment. Did you also take into account the process that it was energy demanding? Those dyes. Can you explain me that, how that works? In light of this, do you? Yeah.
Okay, thank you. The pH rhodos is have the fluorescence labeled with the dyes. So the dyes labeled with myelin, and they will uptake by the cells. When they uptake in the cells is not have the fluorescence. The fluorescence only activated in the lysozyme when the environmental pH lower than 4 that could activate the fluorescence.
Yeah,
that's the answers.
Yeah. Okay.
So
I think we have to close because I will also need to give a final question.
Thank you, Mr. Prorector.
So sorry to disturb you, Dr. Panis. So Ms. Wu, maybe you're a little bit surprised, but we have 5 members in the corona, and I would still like to also address in the final minutes a few questions to you, and I would like to ask my secretary, Professor Prickards, also to notice in our final statement that I also joined the discussion officially. Yes. So I was asked 5 minutes before this meeting to join you. So I listened very carefully to your presentation, and already from that presentation, I could make some questions. So I would like to start first, and we just have one question before the time is over. It's some work which I did myself in the past, which is always nice. And you mentioned that you give your bone marrow stem cells in the cisterna magna. That's correct, in the red. However,
yes.
If I noticed on your slide 15, your needle is not pointing to the cisterna magna, but is pointing to the upper part of the spinal cord. So can you give me a little bit of information? How do you inject into the cisterna magna? Because the cisterna magna is situated under the cerebellum. And basically, you have not pointed towards an area under the cerebellum. So how do you approach the cisterna magna?
No, okay. For the cisterna magna, so when we inject the spinal, when we inject the stem cells, is happened in 1 hour of the spinal cord lesion surgery. So we will open in the neck of the rice and to expose the whole spinal cord, and we will calculate from the cell balance, calculate the bones. So in the
So let me interrupt. I'm going to help you a little bit. Suppose this is your brain, and this is your forebrain. And if you're going to inject into the cisterna magna, you have to come with a needle just under the knot of your skull to enter the cisterna magna. But at the same time, on this side, you started a spinal cord injury. So how do you put your animal, how do you treat it, your animal? Because you really have to open a little bit to have an opening to bring the needle into the cisterna magna. How do you work with that? Did you do it yourself?
Actually, is Yorks do the surgery. The animals will in the dorsal part. With the spinal cord, they will expose there, and the hand will a little fall down of the centering tables, so that will expose the cisterna magna there.
I think the beetle will be there any minute. I can give you much more questions, but that basically has to do with the translation aspect. Bringing bone marrow stem cells in the cisterna magna in a rat is different from what you do in a human being. So how would you do it in a human being? You can answer. You still can answer the question after the
Okay, thank you. For the human beings, we don't inject the bone marrow stem cells and the cisterna magna, but on the puncture on the vertical, the puncture verticals, we do it there. I know it's different as the animals experiment, but the stem cells will migrate with the cell brain spinal fluid, so they will go through this fluid, get into the lesion site. That's different.
Okay, thank you very much. Very well answered. Ms. Siyu, the time appointed for defending your thesis has now officially passed. The degree committee will go now to a different breakout room to discuss the quality of your thesis, not so much because that has already been evaluated, but in particularly the way you have defended your thesis this morning. I requested you and some friends who are virtually available somewhere will wait our return and the results of our deliberations. And I would like to ask Luke or Joshua to bring us to another breakout room. Thank you very much. And we will be back in about 12 to 15 minutes.
Thank you. Thank you so much.
Mr. Prorector.
Luke, is everything going well? Yes.
Yes, Mr. Prorector, everybody is here, I think.
Yes, so we can start. Because I will make a little change in the screen then. Okay. Let me see if the candidate is there too. Yeah. Dear Ms. Siyu Wu, the degree committee here present online has assessed the quality of your thesis and your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of Doctor of Maastricht University. Professor Kramer is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and I invite your supervisor now to take the floor and do all official handling. Professor Kramer, I give you the word.
Thank you, Mr. Prorector. And in accordance with the customs at Maastricht University, I will ask you first the question of scientific integrity to which you have to respond. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I do.
By the authority vested in us by law and in conformity with the decision of the committee here online present, I hereby confer upon you, Siyu Wu, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor affixed with the official seal of the university, which is now shown by the beadle.
Thank you.
Dr. Wu, congratulations.
Now
Thank
we
you
have to,
so much.
I give now the word to Professor May, who will give the laudatio in the name of all supervisors.
Dear candidate, I'm very pleased that, and I'm a little bit proud of you that you were able to lead this to a very successful end, not only shown by the papers that you have published, but also by the nice defense of your thesis. We have been working together 4.5 years, and you were my only PhD student at the time. We were a small group, and so we learned to know each other, although we come from very different cultures. The most important observation that I want to make is that you are an independent and steadfast person who is sure of yourself. And you have worked through all this time, always steadfast and managing many organizational difficulties in a country that is not your language and is not your culture. And you have always mastered all these difficulties. You always kept your coolness. I was always amazed by that. Now, the most important part of a PhD thesis in biology is obviously the work in the laboratory. There are scientists who do this very well, and there are scientists who do this less so. And you have always been excellent. All the techniques that were required, you always mastered them, and you mastered them quickly. A testimony to this is the Myelin Phagotosis assay, which you developed yourself and which was quite successful in the results of 2 papers. Most PhD students during their time encounter a crisis. There may have been difficult moments, but there never was a crisis with you. At least as far as I was able to see, you're always sure of yourself. You never doubted of the eventual success of this work, and so finally it came to be. And this was really something that I admire, your confidence and your perseverance. This project was possible because of the initial initiative of Harry Steinbusch, because of a nice interaction we had between Hans de Munter of Neuroplast, Boris Kramer, and myself in Chengdu. And it was possible because of the generous funding of the Chinese Scholarship Council. Now, the idea of this funding is always that young Chinese students go abroad, get trained, and then come back to their country to apply the knowledge that they've learned in the exterior country. And I think that you have completely fulfilled these objectives to the benefit of everybody, to yourself, to my lab, to Maastricht University, and to the society that has paid you for that. And I think that's a very good accomplishment as well. I'm sure that in your future life as a scientist, as I hope, that you will continue and that you will have this success also. So with my warmest heart, I really congratulate you to that accomplishment.
Thank you.
And you give back the word to me now, officially, yes?
Yes, sorry.
Okay, thank you. Thank you very much. I really have to ask this because I wasn't sure if you finished. So Ms. Siyu Wu, I really would like to congratulate you also on behalf of the Board of Deans of our Maastricht University. I know you were aware of the Chinese Scholarship Council program, and basically in our standards, you are number 56 of the Chinese students who accomplished their thesis. I know we have in Maastricht University a large cohort at the moment of 248 PhD students, all sponsored by the Chinese Scholarship Council, which are you aware of. And I also know that in the contract, it means that you have to return to China now. Is that correct? Because officially, after your thesis, you have to return for at least 2 years to work for the Chinese government somewhere where they decided where you have to go. So I wish you a lot of success in your career. I hope you can continue in spinal cord injury work, because there are a lot of people who will suffer also from spinal cord injury in China. This was your day, and I'm not going to speak to you in Chinese. Also, I'm doing at the moment a course in Chinese language. So this is something different, but I think you had some challenges. And that was already mentioned by Professor May. Of course, you did not work in Maastricht, you worked in Toledo. It's pretty far away. I think you had sometimes the opportunity to visit us here in Maastricht and to learn a little bit and to see the differences. And I think you did today a very great job. You spoke very well. Your answers were excellent. And I think I have to also congratulate you, not only you, but also the promotion team, Professor Kramer and Professor Mai, who really helped you over the years to reach this point. Now, saying this, I'm going to finalize. I'm going to wish also my congratulations to your family, maybe, who are virtually available. I'm not sure. Are you at the moment in China? Yes?
Yes.
In which city are you?
In the Shijiazhuang. It's the capital of Hebei province, which is nearby the Beijing, Peking.
Okay, very nice. So I wish also the family all the best. As a gratitude, our technical staff already made a group picture, which we now call a screenshot. So basically, you can use this as a gratitude. I would like to officially ask the members of your promotion team, and particularly Professor Kramer, who is now in Maastricht, that he would like to come to our office to sign the degree. And we would like to ask him to do this within 1 week. If Professor Mai is also available in Maastricht, he also can join and sign it, because we would like to send it as soon as possible to you in China. Saying this,
I find this on September 27th. I'm in Maastricht. If that is still enough, then I could sign it myself. Otherwise, I can give an electronic signature.
No, we sign the official ones, and I will discuss this afternoon when I will be with the Beadle at the office at Maastricht University. If we can wait till the 27th of September, I assume that should be no problem, but not later, please. Okay. Then, saying this, then we're going to stop here. Enjoy the day. Enjoy the day also in China. Enjoy the day in Toledo and Maastricht, and I officially close this ceremony. Thank you very much, and wish you all the best.